Welcome to class 9 biology children. In our previous classes we have discussed plant tissues and its types, and we have discussed some important points about chapter 2. But now, that means today, we are going to discuss activities of chapter 2, and totally we will conclude chapter 2. Next week onwards we will discuss chapter 3. Okay, if you are having textbooks, open your textbook and observe the activities carefully and try to fill the tables in chapter 2. If you are not having textbooks, please observe the video carefully. Okay children, now I am going to open the textbook of class 9th, chapter 2. Okay children, if you have textbooks, open page number 12. Today we are going to discuss activities of chapter 2, plant tissues. Let us see activity 1. Parts of the plants and their functions. I think in earlier classes you have studied parts of the plant body and their functions. So now we are going to discuss the parts of plant bodies and their functions again. First, serial number. Absorption of water from soil. Which part can absorb water from soil? Roots. By using roots, plants can absorb water from soil. Exchange of gases. Exchange of gases means taking of carbon dioxide and releasing of oxygen. It is done with the help of stomata. Okay, stomata. Photosynthesis. Photosynthesis can be carried out by using leaves. Leaves can perform photosynthesis. Transpiration. Transpiration means loss of water from plant body in the form of water vapor. Do you know plants losses water also? So loss of water from the plant body in the form of water vapor is called transpiration. This transpiration is also done by the stomata. What it indicates? Stomata can perform transpiration. Stomata also performs exchange of gases. Next, reproduction. Reproduction is always done by the flowers. Reproduction is always done by the flowers. This is about our activity 1. In this activity, we have identified 5 parts of the plant body along with their functions. Now we are going to observe the activity 2. Cells in onion peel. In 8th class biology, you have studied cells of onion peel. Which shaped cells are there? Which shaped cells? Observe these. Which shaped cells? Rectangular shaped cells. Rectangular shaped cells we can see in the onion peel. How can we prepare the onion peel slide? Last year you have observed preparation of onion peel cells slide. Okay, take a small piece of onion peel and place it on the slide. First we need to take a small piece of onion peel and we need to place it on slide, and we need to put 1 drop of water and then glycerin. Next we need to cover it with cover slip. Next, after 5 minutes we need to observe it under the electronic microscope. Then we can observe the cells like this manner. If you observe this slide, there is a chance to see one wall presented around the cells. That wall is called cell wall. What is the outermost layer in case of plant cell? Cell wall. Cell wall is the outermost layer in case of plant cell. What do you know about these dotted particles? These dotted particles are called nucleus. Here this cell contains 1 nucleus. This cell also contains 1 nucleus. Each and every cell contains 1 nucleus in it. What do you know about a jelly or fluid like material in the cells? That is called cytoplasm. So in this activity 2 we have observed the cells in onion peel. These cells are arranged in which shape? Rectangular shape. Let us see activity 3. Cells in a leaf peel. So In our previous activity, we have observed cells in onion peel. Now we are going to observe cells in leaf peel. Which leaf? Betel leaf. Betel leaf. Betel means what you know about betel leaf? Tamalapaku. If you observe, all of you see figure 2 betel leaf peel. If you observe the cells in betel leaf, there is a chance to see these cells are arranged very closer when compared to onion peel cell. So these all are cells, these all are cells, these all the cells are having similar structure and similar functions. So these are the cells of betel leaf, these are the cells of betel leaf. Next activity, cells in root tip. Root tip. In our previous activities, activity 3 and activity 2, we have observed the cells in onion peel and cells in leaf peel. Now we are going to observe the cells in root tip. So this activity we have in page number 13. Come to the page number 13. If you have textbooks, open the page number 13. See, here in page number 13, we have figure 3. So first, for this activity, we need to take one onion bulb. Onion bulb means one onion. This part is called onion bulb, but actually we can call it as onion. So onion bulb we need to take. What do you know about it? This is glass tumbler. Okay, glass tumbler. Into that we need to fill it with water. Let's take a glass tumbler, fill it with water. Now take a large sized onion bulb, and keep it over the surface of that glass tumbler. Okay, next wait for 1 week. After 1 week, if you observe the bottom of onion bulb, the roots will grow downwards. Next, after 1 week, you need to remove that onion bulb, and you need to take a small piece from those roots and keep it under the electronic microscope. Then we can observe the cells in the onion root tip like this manner. Okay, so these all are the cells in the onion root tip. Okay, so we can see the cells in the onion root tip. What is the use of this root tip? This root tip is useful to take the water, absorb the water and salts from the soil. Okay, come to the next one. Figure 5. Growth of roots in onion bulb. Here, if we place the onion or a glass tumbler which contains water in it, after 1 week it becomes like this manner. Here there is no chance to see any roots, but after 1 week there is a chance to see growing roots from the onion bulb. Did you understand? So if you cut the small piece from these onion roots, then if you place those roots under the electronic microscope, there is a chance to see root tip structure like this manner. Next, types of plant tissues. Already we have discussed types of plant tissues. How many types are there? 4 types are there. Okay, 4 types are there. 4 types are there. Let me observe what they are. Already in our previous classes I have told you meristematic tissues, dermal tissues, ground tissues, vascular tissues. Meristematic tissues again divided into 3 types: apical meristematic, lateral meristematic, intercalary meristematic. Intercalary meristematic tissues are also called cambium tissues. Next, dermal tissues, ground tissues. Dermal tissues forms outer coverings to the plant body. Ground tissues, already I have told you, ground tissues occupies bulk of the plant body. Ground tissues occupies bulk of the plant body. These ground tissues are divided into 3 types: parenchyma, collenchyma, sclerenchyma. Next, parenchyma again divided into 3 types: chlorenchyma, parenchyma, storage tissue. Vascular tissues are divided into 2 types: xylem, phloem. Okay, so initially how many types are there? Okay, totally how many types of plant tissues are there? 4 types: meristematic, dermal, ground and Vascular tissues. Meristematic tissues again divided into three types: apical, lateral, intercalary. Dermal tissues. Epidermal, mesodermal, endodermal tissues are there. Ground tissues, three types: parenchyma, collenchyma, sclerenchyma. Again parenchyma divided into three types. Vascular tissues divided into two types: xylem and phloem. So this is about the classification of plant tissues. Now we are going to discuss these tissues and we are going to observe the figures of these tissues in our textbook. Look at this figure. Meristematic tissues. Figure 6. LS of shoot tip. So if you take the LS means longitudinal section of shoot tip, there is a chance to see apical meristematic tissues. So this part is called, this part contains apical meristematic tissues. Apical, where do we observe apical meristematic tissues? At the shoot tip and at the root tip. Tip of the plant body contains apical meristematic tissues. So these tissues are useful to increase the growth, nothing but length of the plant body. Next, this is the lateral side. At the lateral side we can see lateral meristematic tissues. This is branching side. Here this is one branch, next this is main branch. So in between the branches there is a chance to see intercalary meristematic tissues. Intercalary meristematic tissues are also called cambium tissues. So this is about the meristematic tissues. Come to activity 6. In this activity 6 we have a table 2. We are going to discuss the table. At the tip, arrangement of the cells. At the tip, shoot tip, root tip. Tips are mainly divided into two types here, shoot tip and root tip. At the tip, which tissues we can see? Meristematic tissues. Root tip also we can see meristematic tissues. At the lateral side of the shoot tip and root tip, we can see lateral side means lateral meristematic tissues. This table 2 is very important. Next, at the point of branching we can see cambium. At the point of branching we can see cambium or intercalary meristematic tissues. Okay. Activity 7. Dermal tissues. How many types of dermal tissues are there? Three types: epidermal, mesodermal and endodermal tissues are there. These all dermal tissues are useful to cover the plant body. So see, in the activity 7, dicot stem tissues. Dicot stem tissues we are going to see. Dicot plants stem tissues we are going to observe. See figure 7, dicot stem. If we observe the dicot stem, it contains meristematic tissues outside. Next, vascular tissues inside. Vascular tissues contains xylem and phloem. So these ball-like structures are called vascular tissues. These are vascular bundles. In these vascular bundles, there is a chance to see xylem and phloem. Xylem, phloem. Like that manner, we can see. This middle part is called ground tissues. This is dermal tissues. Outer layer is called dermal tissues. First dermal tissues we can see. Why these dermal tissues forms outer coverings. Next, meristematic tissues. After that, vascular tissues. After that, ground tissues. So like this manner, these tissues can occupy in the dicot stem. Next, dermal tissues. Rheoleaf dermal tissues. Activity 8. If you observe the rheoleaf under the electronic microscope, there is a chance to see the dermal tissues like this manner. What do you know about this green colored one? This green colored one in a rheoleaf dermal tissue is stomata. If you observe the stomata, it contains two kidney or bean shaped structures. These are called guard cells. These are called guard cells. This is one guard cell. Next, this is another guard cells. Okay. Next, what do you know about these? These are the cell walls. These are the cell walls around the stomata. By opening and closing of stomata, what will happen? Exchange of gases. Next, transpiration can be done in the plant body. Next, we are going to discuss ground tissues and its types. Ground tissues occupies bulk of the plant body. Already we know it. Let us see ground tissues types. Parenchyma, soft cells with intracellular spaces are there. Next, collenchyma, some hard cells are there when compared to parenchyma, and intercellular gaps are low when compared to parenchyma. Sclerenchyma, very toughest cells we can see with the absence of intercellular gaps. Already we have discussed about these tissues in our previous classes. Let us see parenchyma types. Parenchyma divided into three types. Again, let us see. If parenchyma cells contains chloroplast in it, then it is called chlorenchyma. This chlorenchyma is useful to perform the photosynthesis. If parenchyma cells contains air spaces in it, then it is called aerenchyma. It is useful to make them to float the plant in water. If parenchyma stores water and food materials, then it is called storage tissues. Okay, so these are the types of ground tissues. Who gave the term parenchyma? Already I told you. Who gave the term parenchyma? Nehemiah Grew. He wrote a book, Anatomy of Plants, in the year 1682. Which book he wrote? Anatomy of Plants, in the year 1682. Who gave the term parenchyma for the first time? Nehemiah Grew. Nehemiah. Nehemiah. Nehemiah Grew. 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 Nehemiah Grew. Very famous scientist. Vascular tissues. Vascular tissues are also called transportation tissues. Why does these tissues are called transportation tissues? These tissues are useful for transportation of water and minerals. Okay, that is why these tissues are called transportation tissues. Let us see. Here in figure 12 there is a chance to see xylem and phloem. So these are the cells of xylem. These are the cells of phloem. Okay. So in case of xylem there is a chance to see vessels. Next tracheids. Next fibers. With the help of these vessels, tracheids and fibers, xylem can transport water. Next, with the help of sieve cells, next sieve fibers and sieve vessels and companion cells are also there in phloem. With the help of these oval cells, what will happen? Phloem can transport food materials from one place to another place. Let us see figure 11, TS of stem. It is very important diagram from chapter 2. In our next class or while explaining the chapter 3, we will draw the diagram. TS of stem. Observe this. So if you cut the stem, there is a chance to see xylem bundles, some bundles like structures inside of the stem. So this is one bundle, next this is also another bundle, next bundle, next bundle, next bundle, like this manner. So these bundles are called vascular bundles. In these vascular bundles, there is a chance to see here in pink color, and also you can see black color dot like structures. So pink color dot like structures are called xylem. Next black colored dot like structures are called phloem. So xylem is useful for transportation of water and minerals. Here phloem is useful for transportation of food materials. So this is about the structure of stem or TS of stem. So with these activities, our chapter 2 is completed. In our next class onwards, we will discuss chapter 3 and we will draw the diagram of this TS of stem. Okay children, thank you.